Welcome to the Deep Dive. This is where we really get into the weeds on fascinating, complex topics, pulling out the key insights for you. Today, we're diving deep into an organ that, well, Homer W. Smith, a real giant in renal physiology, described it incredibly well. He said, the kidney presents in the highest degree the phenomena of sensibility, the power of reacting in a direction which is appropriate for the survival of the organism, a power of adaptation which almost gives one the idea that its component parts must be endowed with intelligence. We're talking about the human kidney, and it's so much more than just a filter. It's a master regulator, a biological marvel, really. The level of detail is just astounding. Our mission for you listening is to really help you master this material, like the elite professionals in the field do.
That Smith quote, yeah, it just nails it, doesn't it? The kidney isn't simply filtering waste. It's this dynamic, almost intelligent regulator. Its core functions are absolutely fundamentally vital for everything. First off, and maybe the most critical, it meticulously manages your body fluid osmolality and volumes. Now, osmolality, that's basically the concentration, of all the dissolved stuff, salts, glucose, urea in your body fluids. Keeping that balance right is crucial. It stops your cells from swelling up or shrinking, which would, well, disrupt everything. And it also directly impacts your cardiovascular system, your blood pressure. Huge. Second, the kidneys are like the ultimate guardians of your electrolyte balance. We're talking essential ions here: sodium, potassium, chloride, bicarbonate, hydrogen, calcium, phosphate. The list goes on. For your body to stay balanced, what you take in each day has to equal what you excrete. It's simple math, really. If you take in more than you get rid of, that's a positive balance. The total amount in your body goes up. If you excrete more than you take in, negative balance. And for many of these critical electrolytes, the kidneys are the main way out, sometimes the only way out. Third, they're central to keeping your acid-base balance in check. Almost every metabolic process in your body is incredibly sensitive to pH, that measure of acidity. Even tiny shifts can be, well, catastrophic. The kidneys work together in coordination with your lungs and liver to keep your blood pH locked into a very, very narrow, safe zone. Fourth, they just tirelessly get rid of metabolic waste products. Things like urea, which comes from breaking down proteins and amino acids, uric acid from nucleic acids, creatinine from muscle metabolism, plus other stuff from hemoglobin breakdown, hormone leftovers, you name it. They make sure these potentially harmful things don't build up to toxic levels. And it's not just internal waste, they're your detox system too, clearing out foreign stuff like drugs, pesticides, other environmental toxins. And finally, a role that often gets overlooked but is profoundly important: the kidneys are endocrine glands. They actually make and secrete vital hormones. There's renin. That's an enzyme that kicks off the whole renin-angiotensin-aldosterone system.
Right, the RAAS system, big for blood pressure.
Exactly. Absolutely key for regulating blood pressure and your sodium and potassium balance. Then there's calcitriol. That's the active form of vitamin D3. This hormone is essential for absorbing calcium from your food and getting it properly deposited into your bones. When kidney function is impaired, like in kidney disease, calcitriol production drops. That leads to less calcium absorption, contributing to the bone problems we often see in chronic kidney disease.
Ah, okay. That makes sense.
And don't forget erythropoietin, EPO. This hormone tells your bone marrow to make red blood cells. A big drop in EPO production is exactly why anemia, a shortage of red blood cells, is such a common and serious issue in chronic kidney disease.
It's just incredible. So many critical jobs packed into these relatively small organs. Yeah. And when they start to fail, the consequences are just huge. This isn't just abstract biology, is it? It's a massive public health crisis. We're talking, what, over 37 million people in the U.S. alone with kidney disease? That's 1 in 7 adults. And here's the really scary part, the thing that just, wow, blew my mind. Chronic kidney disease, CKD, it's massively underrecognized. Something like 90% of people with CKD don't even realize they have it.
90%, it's staggering.
Think about that. Half of the people with really low kidney function don't know. It's a silent epidemic, truly. And the numbers just get worse. CKD is the 8th leading cause of death in the U.S. Over 100,000 deaths every year. The cost, unbelievable. Over $70 billion a year just for CKD care and Medicare. And another $50 billion for those who progress to end-stage kidney disease, ESKD. Immense figures. Right now, over 786,000 Americans are living with ESKD. The vast majority, 71%, are on dialysis. 29% have a transplant. And the main drivers: diabetes and hypertension, high blood pressure. Even with over 23,000 kidney transplants happening each year, there's still over 90,000 people on the waiting list, hoping. And these organs are just indispensable, literally life-saving. Okay, given all that weight, how do we even begin to wrap our heads around these incredible organs? Let's start big picture. Where are they? What do they look like, macroscopically?
That's definitely the place to start because their location, their structure, it's all tied into how they work. So you have two kidneys, a pair, tucked away deep in your abdomen. Think about your lower back on either side of your spine. They're nestled against the back wall of your abdomen behind the peritoneum. That's the lining of your abdominal cavity. In an adult, each one is roughly the size of, say, a bar of soap. Weighs about 115 to 170 grams. Dimensions are around 11 centimeters long, 6 wide, and 3 thick. Now, if you were to slice one open, lengthwise, you'd immediately see two main regions. There's an outer layer, lighter in color, called the cortex, and an inner, darker region, the medulla. In humans, the medulla is really neatly organized into these cone-shaped structures like little pyramids. We call them renal pyramids. The base of each pyramid sits right at the border between the cortex and medulla, and its pointy tip, called a papilla, sticks into a little cup-shaped collector called a minor calyx.
Okay, like a funnel.
Exactly, like tiny funnels. They collect the urine as it drips off the papilla. Then, several of these minor calyces merge together to form bigger pouches, the major calyces, usually 2 or 3 of those, and all the major calyces drain into the renal pelvis. That's the widened, funnel-shaped top part of the ureter. The ureter is the tube carrying urine from the kidney down to the bladder, and the balls of the calyces, the pelvis, the ureters, they all have smooth muscle. It contracts rhythmically, pushing the urine along, like a little conveyor belt.
So 2 incredibly busy, complex organs tucked away back there. But you mentioned something earlier about their blood supply that sounded just extraordinary. The volume of blood.
Oh, absolutely. It's one of the most astonishing things about them. Together, your kidneys get about 25% of your total cardiac output when you're resting.
A quarter. That's huge.
It's staggering. 1.25 liters of blood every single minute. And remember, the kidneys themselves make up less than 0.5% of your total body weight, but they demand a quarter of the blood your heart pumps. This massive flow is absolutely critical for all that filtering and regulating they do. Blood comes in through the renal artery, which then branches out like a tree, getting smaller and smaller. Interlobar arteries, then arcuate arteries, interlobular arteries. And finally, these tiny vessels called afferent arterioles. These afferent arterioles lead straight into the glomerular capillaries, the main filtering units. Now, here's a really unique design feature. Instead of going right into a vein, these glomerular capillaries actually come back together to form another arteriole, the efferent arteriole.
Wait, an arteriole after the capillaries? That's unusual.
Highly unusual. And this efferent arteriole immediately branches out again into a second, much larger capillary network, the peritubular capillaries. These wrap all around the rest of the functional units, the nephrons, providing them blood and picking up all the stuff the kidney reabsorbs. Then the venous system pretty much mirrors the arteries in reverse. Blood drains from the peritubular capillaries into interlobular veins, arcuate veins, interlobar veins, eventually converging into the big renal vein, which carries the cleaned blood back into your main circulation.
The dual capillary system, that's really clever. The 2 processing stages. Okay, but what about those functional units you mentioned, the nephrons? You said millions of them. What is a nephron exactly? How does it do its microscopic magic?
Right, the nephron, that's the real microarchitect, the absolute workhorse of the kidney. Each kidney packs in about 1.2 million of them, give or take. And picture each one not as a solid thing, but as this incredibly complex, long, hollow tube made of just a single layer of specialized epithelial cells. Every nephron kicks off with the renal corpuscle. Think of it as the kidney's initial blood cleaning station. It's got that dense little knot of glomerular capillaries we just talked about, all tucked inside a cup-shaped thing called Bowman's capsule. Fluid filters out there into the capsule and then enters the proximal tubule. This bit first coils up quite a bit, then straightens out and dives down towards the medulla. After the proximal tubule comes the famous loop of Renal, that U-shaped part, absolutely crucial for concentrating urine. It has a descending thin limb, makes a sharp hairpin turn, then an ascending thin limb, though only in some nephrons, and then a thick ascending limb. Near the end of that thick ascending limb, there's a really special little patch of cells called the macula densa.
Macula densa, okay.
And here's the clever bit. This macula densa actually loops back and sits right next to the afferent and efferent arterioles of its own glomerulus. It's a direct communication line.
Wow. Built-in feedback.
Exactly. Then, beyond the macula densa, the fluid flows into the distal tubule, which eventually connects to a cortical collecting duct. These collecting ducts then merge together, becoming outer medullary, then inner medullary collecting ducts, funneling the final urine down towards the renal pelvis.
So it's this long, winding journey through different specialized zones. How are the cells themselves specialized for all these different jobs? What's happening right down at the cellular level?
Great question. Because the cells lining each part are totally unique, tailored for their specific function. Look at the proximal tubule cells. On the side facing the fluid, the apical side, they have this massive brush border. It's like a dense forest of tiny finger-like projections, massively increasing the surface area, perfect for reabsorbing huge amounts of stuff.
More surface area, more absorption. Makes sense.
Right. And then on the other side, facing the basolateral side, their membrane is deeply folded, and they are absolutely jam-packed with mitochondria. Mitochondria are the cell's power plants, remember. So this tells you these cells are incredibly active, using tons of energy for active transport, pumping things across. Now contrast that with the thin rings of the loop of Henle. Their cells have much simpler surfaces, very few mitochondria, suggests a more passive role, less energy-intensive transport. But then you get to the thick ascending limb and then distal tubule, and boom, lots of mitochondria again, extensive folds on the basolateral side, back to heavy-duty active transport. Even the collecting duct isn't uniform. It has different cell types. You've got principal cells. They have moderately folded basolateral membranes, fewer mitochondria. Their main job is fine-tuning sodium chloride reabsorption and potassium secretion. Really important for final adjustments. Then you have intercalated cells. These guys stand out because they're loaded with mitochondria. They're the acid-base specialists. One type pumps out hydrogen ions and reabsorbs bicarbonate, fights acidosis. Another type does the opposite. Secreting bicarbonate helps with alkalosis and can also reabsorb sodium chloride.
So specialized cells for different pH problems.
Precisely. And finally, way down in the intermedullary collecting duct, the cells become less specialized again. Simpler surfaces, fewer mitochondria.
That's an incredible level of cellular detail.
Yeah.
You also mentioned something about tiny antennae on these cells. Primary cilia? What are those doing there?
Ah, yes, the primary cilia. Fascinating little structures. Pretty much all the cells along the nephron tubule, except for those intercalated cells, have a single non-moving cilium, like a tiny antenna sticking out into the fluid from their top surface. And they're not just decorations, they're incredibly sophisticated sensors. They act as mechanosensors. They literally feel the flow rate of the fluid rushing past. And they're also chemosensors detecting specific chemicals in that fluid.
Sensing flow and chemicals.
Yeah. And crucially, when they sense something, a change in flow, a specific molecule, they trigger calcium signals inside the cell. These calcium signals are vital. They control all sorts of things: kidney cell function, growth, specialization, even program cell death, apoptosis. Now, digging deeper, specific proteins make these cilia work. Polycystin 1 from the PKD1 gene and polycystin 2 from the PKD2 gene. These proteins sit right there in the cilium's membrane, and they basically form a channel that lets calcium ions into the cell. They play a critical role, for example, in how principal cells secrete potassium in response to flow. Here's how it works. More fluid flows past, the cilium bends, like flicking a switch. This bending activates that PKD1, PKD2 complex, the calcium channel. Gate opens, more calcium flows into the cell. This rise in internal calcium then activates potassium channels on the cell surface, boosting potassium secretion into the urine. It's a really elegant feedback loop. And this brings us straight to a major clinical issue: autosomal dominant polycystic kidney disease, ADPKD. It's the most common inherited kidney disease out there. It affects maybe 1 in 1,000 people, over 12.5 million worldwide. And it's mostly caused by mutations in that PKD1 gene, 85 to 90% of cases, or the PKD2 gene is about 15%.
The same genes coding for those cilia proteins.
Exactly the same ones. The main feature of ADPKD is that the kidneys get massively enlarged, filled with hundreds, sometimes thousands, of fluid-filled cysts. These cysts can get huge, like 20 centimeters across, just destroying normal kidney tissue. Cysts can pop up in the liver too. Sadly, about half of ADPKD patients end up needing dialysis or a transplant by age 60, end-stage kidney disease. And the underlying cause is thought to be exactly these defects in calcium entry through the faulty polycystin channels. It messes up those crucial calcium signals that normally control cell growth and specialization in the kidney.
Wow. So a tiny flaw in those cellular antennae leads to such a devastating disease. That really hammers home that intelligence quote. You also mentioned that not all nephrons are the same, different types.
That's right. They're not all identical. Two main flavors: superficial nephrons and juxtamedullary nephrons. You have about 10 superficial ones for every one juxtamedullary. Superficial nephrons, their glomerulus is closer to the kidney surface in the outer cortex. Their loops of Henle are shorter, some barely dip into the medulla. Juxtamedullary nephrons, though, their glomerulus is deep in the cortex, right next to the medulla, hence the name juxta, meaning near. And they have two key differences. First, their loop of Henle is much longer, plunging way down deep into the medulla. Second, their efferent arteriole, remember that vessel leaving the glomerulus. It forms not just the usual peritubular capillaries, but also this specialized network of straight vessels called the vasa recta. The vasa recta are really interesting. They run straight down into the medulla, parallel to the long loops of Henle and the collecting ducts, form intricate capillary networks around them. Blood then flows back up towards the cortex through ascending vasa recta. Now, even though these vasa recta only carry a tiny fraction of the total renal blood flow, less than 0.7%, they're absolutely vital for the medulla. They're the supply lines, bringing oxygen and nutrients down deep. They deliver stuff for secretion into the tubules. Crucially, they carry away the water, and still it's reabsorbed from the medulla, getting them back into circulation. And they are absolutely fundamental to the kidney's ability to concentrate or dilute urine. A whole other amazing story there.
That's just incredible precision, even down to different nephron types and specialized blood vessels. Okay, let's shift gears slightly. The very first step in making urine, ultrafiltration. You said this is passive. How does the kidney make sure only the right stuff gets filtered out of the blood?
Right, ultrafiltration. It's the passive movement of fluid, basically plasma, but with very little protein out of the glomerular capillaries and into that Bowman's space, like a high-pressure filter press. And the structure enabling this is the glomerulus itself, that bundle of capillaries fed by the afferent arteriole, drained by the afferent. During development, these capillaries essentially push into the end of the forming tubule, creating Bowman's capsule around them. Bowman's capsule has two layers, an outer parietal layer, just thin epithelial cells, and an inner visceral layer directly touching the capillaries. This inner layer is made of those highly specialized cells called podocytes.
Podocytes, foot cells.
Exactly. They have these intricate foot-like processes that wrap around the capillaries, interlocking with each other. The space between these two layers is Bowman's space. At one end, the urinary pole, it leads into the proximal tubule. The other end, the vascular pole, is where the afferent and efferent arterioles connect.
So it's not just a simple sieve then. There must be a really sophisticated barrier there to be so selective. What are the layers of this glomerular filtration barrier? How do they work?
You got it. Far from simple. It's a highly specialized three-layered barrier. Layer 1. The capillary endothelium itself. The inner lining of the glomerular capillaries. It's fenestrated full of tiny holes, or windows, about 700 angstroms across. Super tiny. These holes let water, small solutes like sodium, glucose, urea, even small proteins pass through easily. But they block the big stuff. Red cells, white cells, platelets, no way through. Plus, the surface of these endothelial cells is coated with negatively charged glycoproteins. Think of them as electrostatic shields. They repel negatively charged molecules, especially albumin, the main protein in blood, which is also negatively charged. And these endothelial cells aren't just passive. They make signaling molecules too, like nitric oxide, a vasodilator, and endothelin-1, a vasoconstrictor, helping control local blood flow. Layer 2, the glomerular basement membrane. This is like a porous, gel-like mesh made of special proteins. Type IV collagen, laminin, negatively charged proteoglycans like agrin and perlecan. It acts as another size filter, but maybe more importantly, it's a strong charge barrier because of all those negative charges woven into it. Layer 3, the podocyte foot processes. Those interdigitating fingers from the podocytes covering the basement membrane. They interlock but leave these tiny gaps between them called filtration slits. And spanning across these slits is an incredibly thin membrane like a final screen called the filtration slit diaphragm. And has even tinier pores, maybe 40 by 140 angstroms. And it's made of crucial proteins, nephron, NEPH1, podeucin, plus others inside the cell that anchor it, like icactin-4, CD2AP. This slit diaphragm is the ultimate size-selective filter. It makes sure hardly any proteins or other large molecules that might have somehow squeezed past the first two layers actually get into Bowman's space.
So a multi-stage security system, checking both size and charge. That's incredibly sophisticated.
Right.
How does that end up dictating what actually makes it into the filtrate?
Precisely. The final product, the ultrafiltrate, is a direct result of that dual restriction, size and charge. For neutral molecules, if they're smaller than about 18 angstroms radius, they zip right through, freely filtered. If they're bigger than 42 angstroms, forget it, not filtered. In between 18 and 42, they get filtered to varying degrees depending on their exact size, but charge is just as critical, maybe even more so. Remember, all three layers of the barrier are studded with negative charges, so they actively repel other negatively charged molecules. And most plasma proteins, especially albumin, are negatively charged.
So that's why albumin mostly stays in the blood.
Exactly. Albumin is fairly largish, 35.5 angstroms radius, but its strong negative charge is the main reason it's poorly filtered. It gets repelled. And the tiny amount of albumin that does sneak through usually gets immediately gobbled up, reabsorbed by the proximal tubule cells. That's why healthy urine has virtually no albumin. We can show this really clearly using dextran's neutral sugar polymers. If you compare how negatively charged neutral and positively charged dextrans of the same size get filtered, you see the negatively charged ones are blocked much more effectively. It beautifully proves the importance of charge selectivity. And there's one more player in the renal corpuscle, the mesangium. It's made of mesangial cells and the matrix they produce, sitting in the middle of the glomerular tuft. Mesangial cells are interesting. They're a bit like smooth muscle cells. They provide structural support for the capillaries. They also make extracellular matrix, act like little vacuum cleaners, phagocytosis, removing trapped molecules, and secrete signaling molecules like prostaglandins and cytokines. Because they can contract, and they're right there among the capillaries, they might even influence GFR by tweaking blood flow or the filtering surface area. There are also extraglomerular mesangial cells located just outside, between the afferent and efferent arterioles.
Okay, so if this incredibly fine-tuned barrier gets damaged,
Yeah.
what tells us something's wrong? What are the clinical signs?
Yeah, that's a critical clinical question. When you start seeing significant amounts of protein in the urine proteinuria, that's a major red flag. The barrier's compromised. If lots of protein, especially albumin, leaks out into the urine, your blood albumin levels drop, hypoalbuminemia. Albumin normally helps keep fluid inside your blood vessels. When it's low, fluid leaks out into your tissues, causing swelling all over, generalized edema. That trio, massive proteinuria, low blood albumin, and widespread edema, that's the classic picture of nephrotic syndrome. Now, nephrotic syndrome can happen for different reasons. It could be genetic mutations in those genes coding for key barrier proteins we talked about, like nephrin and PHS1, pidosin and PHS2, or those anchoring proteins like iaptinin-4, ACTN4.
So inheriting a faulty barrier protein.
Exactly. Mutations in the nephrin gene cause a severe form right from birth, congenital nephrotic syndrome, massive protein loss, kidney failure very early. Pidosin mutations also cause nephrotic syndrome, but it's not always genetic. It can also be triggered by the immune system going awry, maybe after an infection, and attacking the filtration barrier. Another important condition related to barrier problems is Alport syndrome. This is genetic too, affecting the type 5e collagen in the glomerular basement membrane, that second layer. It leads to structural problems there, causing blood in the urine, hematuria, protein in the urine, proteinuria, and usually a slow decline in kidney function over time. Alport's accounts for maybe 3% of childhood CKD and a smaller fraction of adult end-stage disease. It's caused by mutations in the COL4A3, COL4A4, or COL4A5 genes. Most often, it's X-linked from COL4A5 mutations, so it hits males harder. But you can get autosomal forms from COL4A3 or COL4A4 mutations too. And actually, with newer genetic testing, we're realizing these COL4A mutations might be more common than we used to think.
So the barrier is incredibly complex, and damage has huge consequences. But how do the kidneys manage to keep their own blood flow and filtration so incredibly stable? Despite blood pressure going up and down all day, it seems like they have their own internal control system.
They absolutely do. It's a marvel of physiological engineering. Renal blood flow, RBF, does more than just supply the kidney, remember? It determines the GFR, influences reabsorption in the proximal tubule, helps concentrate urine, delivers oxygen and nutrients, takes away waste and reabsorbed fluid. It's critical. And basic physics says flow equals pressure divided by resistance. The key resistance points in the kidney are those afferent and efferent arterioles and the interlobular arteries. The phenomenon you're describing is called autoregulation. It's the kidney's amazing ability to keep RBF and GFR remarkably constant, even when your main arterial blood pressure swings between about 90 and 180 millimole Hg.
That's a huge range.
It is. And this stability is mostly achieved by precisely adjusting the resistance of the afferent arteriole, the inflow pipe. Since both RBF and GFR are kept steady across this range, and RBF heavily influences GFR, it makes sense the same mechanisms control both. There are two main mechanisms behind this autoregulation. First, the myogenic mechanism. This is built right into the smooth muscle cells of the afferent arterial wall. It's pressure sensitive. When arterial pressure rises, it stretches the arterial wall. The smooth muscle cells automatically contract in response to that stretch. This contraction increases resistance, counteracting the pressure rise, and helps keep blood flow and thus GFR stable, like a reflex in the vessel wall itself. The second mechanism is tubuloglomerular feedback, PGF. This is that incredibly elegant salt-sensing feedback loop involving the macula densa, part of the juxtaglomerular apparatus.
Ah, the JGA. We mentioned that structure near the glomerulus.
Exactly. Here's how TGF works. Let's say your GFR goes up for some reason. More fluid filters through. This means more sodium chloride flows past the macula densa cells in the thick ascending limb. These cells are super sensitive to this. They detect the increased salt by taking more of it up through a special transporter called NKCC2. This increased salt uptake triggers the macula densa cells to release signaling molecules, specifically ATP and its breakdown product, adenosine. These molecules, ATP and adenosine, then act as local messengers, causing the nearby afferent arteriole to constrict. ATP hits P2X receptors, adenosine hits A1 receptors, both on the arteriole's smooth muscle cells. Both signals cause calcium levels inside those muscle cells to rise, making them contract. Vasoconstriction. This narrows the afferent arteriole, reducing blood flow into the glomerulus, and bingo, GFR comes back down to normal.
Wow. A self-correcting loop based on salt concentration.
Precisely. And it works the other way, too. If GFR drops, less salt reaches the macula densa, less ATP-adenosine is released, the afferent arteriole relaxes, dilates, and GFR comes back up. It's remarkably precise.
So the macula densa is like the kidney's internal thermostat, adjusting things based on the flow composition.
Yeah.
But it's not just a simple on-off switch. Right?
Yeah.
You mentioned other factors involved.
Exactly right. It's more nuanced. While ATP and adenosine are the key constrictor signals here, other molecules modulate TGF. Nitric oxide, NO, a vasodilator, is also produced by the macula densa, and it actually dampens the TGF response, helps fine-tune it, prevent overcorrection. On the flip side, angiotensin II enhances the TGF response, makes it stronger. So the macula densa is constantly balancing these constrictors and dilators for really exquisite control. Plus, the macula densa also controls renin release from nearby granular cells. Less salt arriving stimulates renin release, linking this local feedback to the body's main blood pressure system. And that role of adenosine and TGF is really fascinating, cellularly speaking, because usually, elsewhere in the body, adenosine dilates blood vessels.
So here it constricts.
Here it constricts the afferent arteriole. It's crucial for TGF. We know this because if you take mice that genetically lack the A1 adenosine receptor, they completely lose their TGF response. Gone. When GFR rises, more salt enters the macula densa via NKCC2. This triggers ATP release through special channels. Adenosine production also goes up. Both signals hit the efferent arteriole muscle cells, raise calcium, cause constriction, and lower GFR. It's a beautifully coordinated cellular event. The physiological importance of all this autoregulation is huge, absolutely vital for survival. It stops your body from wildly losing or retaining massive amounts of fluid and salt every time your blood pressure fluctuates a bit. Imagine peeing gallons every time you stood up quickly. Autoregulation prevents that chaos, keeping your internal environment stable. But, like any system, it has limits. One, it fails if your blood pressure drops too low, below about 90 mmHg. That's a critical threshold. Two, it's not absolutely perfect. RBF and GFR do still wiggle slightly with pressure changes, even within the autoregulatory range. And three, this powerful internal system can be overridden or strongly influenced by hormones and sympathetic nerves, especially when your body's fluid volume changes dramatically, think dehydration or major bleeding.
It's incredible. A built-in stabilization system. But you just said hormones and nerves can override it. So what happens in those more extreme situations? When the body needs to take charge?
Right. Connecting to the bigger picture, even with autoregulation humming along, external factors can take the reins, especially under stress. The basic principle is still modulating the resistance of those afferent and efferent arterioles, but now via hormones and nerve signals. Just quickly recap. Constrict the afferent inflow, lower GFR. Constrict the efferent outflow can initially raise GFR, but high levels lower RBF GFR. Dilate either opposite effects. Let's look at sympathetic nerves first. Part of your fight or flight response, they innervate both afferent and efferent arterioles. Normally, when you're well hydrated, sympathetic activity to the kidneys is low. But if your body fluid volume drops, or during intense stress like fear or pain, the sympathetic system fires up. Nerves release norepinephrine. Your adrenals release epinephrine and dopamine. These stress hormones hit alpha-1 receptors, mostly on the afferent arterioles, causing them to clamp down. Vasoconstriction. Result: a big drop in both renal blood flow and GFR. Think about a really severe situation like hemorrhage, losing a lot of blood.
Okay, like a trauma situation.
Exactly. ECF volume plummets, blood pressure drops. Your body's pressure sensors, baroreceptors, detect this and trigger intense sympathetic firing to the kidneys. Norepinephrine causes powerful constriction of both afferent and efferent arterioles. RBF and GFR tank. Why? Because this intense resistance increase in the kidneys and elsewhere helps jack up your overall peripheral resistance, fighting the drop in blood pressure. Your body prioritizes maintaining systemic blood pressure to keep vital organs perfused, even if it means temporarily sacrificing normal kidney function. Survival mode. Now, angiotensin II, A-THU, another major player. Produced systemically and locally in the kidney, it constricts both afferent and efferent arterioles, generally lowering RBF and GFR. But here's that nuance again. The efferent arteriole is more sensitive to A2, so at low A levels, efferent constriction wins out. This actually bumps up pressure inside the glomerulus, potentially increasing GFR slightly, even as overall RBF falls.
Interesting trade-off.
Right. But at high A levels, both arterioles constrict hard, dropping both GFR and RBF. ADI works hand in glove with the sympathetic system during emergencies like hemorrhage to cut kidney blood flow, raise systemic pressure, and conserve fluid. What about prostaglandins, PGE1, PGE2, PGA2? Normally, in healthy folks at rest, they're not major players in RBF control, but they become absolutely critical during stress states, hemorrhage, low fluid volume. In these situations, they're produced locally in the kidney and act as protectors. They increase RBF, often without changing GFR much, by counteracting the strong vasoconstriction from sympathetic nerves and A time.
So they prevent the kidney from shutting down completely.
Exactly. They prevent severe renal ischemia, lack of blood flow, which could cause serious damage. Interestingly, as people get older, prostaglandins become more important for maintaining baseline RDF-GFR. That's why NSAIDs, nonsteroidal anti-inflammatory drugs like ibuprofen, which block prostaglandin production, can be risky, especially for the elderly. They can cause a significant drop in RBF and GFR, sometimes triggering acute kidney injury. Nitric oxide, NO. We mentioned it briefly. It's a key vasodilator produced by the endothelial cells lining the arterioles. Works under normal baseline conditions. It helps balance out the constricting effects of AFib and sympathetic nerves. Things that stimulate NO production include the sheer force of blood flow itself and various hormones like acetylcholine, histamine, bradykinin, ATP. More NO means dilation of both afferent and efferent arterioles, leading to increased RBF and GFR. Clinically, no pathways can go wrong. In diabetes, for example, sometimes there's excess renal NO production. This might contribute to glomerular hyperfiltration GFR being too high, which paradoxically can cause long-term damage. In some types of hypertension, the kidneys might not produce enough NO in response to high salt intake, which normally helps keep blood pressure down. This failure contributes to the high blood pressure. Then there's endothelin, a really potent vasoconstrictor, made by endothelial cells, mesangial cells, some tubule cells, stimulated by things like A3NO, epinephrine, shear stress. Endothelin clamps down hard on both afferent and efferent arterioles, dropping GFR and RBF significantly. While its role in normal resting state might be minor, endothelin levels are often elevated in various kidney diseases, like diabetic kidney disease, suggesting it plays a role in the pathology. Bradykinin, generally a vasodilator, made locally when an enzyme called kallikrein acts on kinagen. Bradykinin works mostly by triggering NO and prostaglandin release. Net effect: increased RBF and GFR. Natriuretic peptides AMP from the heart's atria, BNP from the ventricles, released when your fluid volume expands and stretches the heart walls. They act to dilate the afferent arteriole and constrict the efferent. Result: a nice little boost in GFR without much change in RBF, helping you excrete that excess fluid. ATP adenosine triphosphate can actually do both things. Released locally, it can constrict the afferent arteriole, part of that TGF mechanism, lowering RBF-GFR. But it can also stimulate NO production, doing the opposite, increasing RDF-GFR. Context matters. Just a few more to show the breadth of control. Glucocorticoids, like therapeutic steroids, tend to increase GFR and RBF. Histamine, released locally, especially during inflammation, tends to decrease resistance in both arterioles, raising RBF without necessarily changing GFR much. And dopamine, actually produced by the proximal tubule, acts as a vasodilator, increasing RBF and inhibiting renin release. So if you pull it all together, those endothelial cells lining the renal arterioles are major control hubs. They produce this whole cocktail of local hormones, NO, prostacyclin, PGI2, endothelin, A, that fine-tune the resistance of the arterioles. Shear stress, various hormones trigger NO release, vasodilation. ACE enzyme on the endothelin makes AE, vasoconstriction. Sympathetic nerves and ADH trigger PGI2, PGE2 release, vasodilation, protective. Endothelin release causes vasoconstriction. It's a constant balancing act. And clinically, this is exactly what we target with blood pressure meds. ACE inhibitors, lisinopril and olapril. ACE normally does two things: makes ACE a constrictor and breaks down bradykinin dilator. So ACE inhibitors block both those actions.
Well, they block the enzyme, so you get less AI and more bradykinin. Both effects lower blood pressure systemically and, importantly, lower resistance in the kidney, often increasing GFR and RBF. The other main class, angiotensin II receptor blockers, ARBs, like losartan, they work differently. They just block AI from binding to its main receptor, AT1. So they block A's constrictor effects, particularly on the efferent arteriole, increasing RBF and GFR. But unlike ACE inhibitors, ARBs don't affect bradykinin levels. It's a more specific AI blockade. Understanding these pathways is key to using these drugs effectively.
We have covered an incredible amount today. Just amazing. From the big picture anatomy of the location, that massive blood supply, right down to the nitty-gritty cellular details, the molecular gatekeepers of the filtration barrier, and this incredibly complex web of hormones and nerves constantly adjusting kidney function. It's really stunning how these small organs, less than 0.5% of body weight, can handle a quarter of our blood flow, meticulously managing fluids, electrolytes, acid-base, waste, plus making hormones. The sheer elegance is breathtaking. That multi-layered filter, the self-correcting TGF loop, you really see why Homer Smith used words like sensibility and intelligence.
Absolutely. And what we really hope you take away today is how interconnected it all is. To truly master this, to think like an elite professional, you have to appreciate how a tiny change at the molecular level, a single gene flaw affecting a ciliary protein, like an ADPKD, can ripple outwards, impacting the whole organ, the whole person's health, the kidney's adaptive power, its knack for always reacting appropriately for survival. It really does feel like something bordering on intelligence, as Smith suggested. It's just a masterpiece of biological engineering. And maybe that leaves us with a final thought for you, the listener. We've dived deep into the complexity, the delicate balance needed just for these organs to work. What does this intricate biological intelligence suggest about the resilience or maybe the hidden fragility of any complex system?
Right.
Whether it's biological, ecological, maybe even societal, what happens when just one critical component starts to malfunction? Something to ponder.